Before a novel oral solid dosage product can reach a patient, it must pass through a strict, legally binding gatekeeper. This document is the specification table. The International Council for Harmonization, through its Q6A guideline, defines a specification in exact terms. It is a targeted list of tests, analytical procedures, and absolute numerical acceptance criteria. When scaling up the synthesis of a novel chemical entity, manufacturers are wrangling millions of discrete molecules into a highly ordered physical state. The inherent chemical complexity of these compounds necessitates stringent binding controls to guarantee uniformity across commercial batches. Conformance to these specifications does not mean conducting an exhaustive scientific characterization of the molecule on every single batch. Rather, it is a targeted confirmation. The manufacturer focuses only on those specific characteristics proven to ensure the safety and efficacy of the drug. The specification cannot guarantee quality on its own. It acts as the capstone within a total control strategy, relying entirely on the foundational pillars of good manufacturing practices, process validation, and in-process controls. Without those systems, the final specification tests are meaningless. To understand how a pharmaceutical manufacturer legally justifies these precise limits to global regulatory agencies, we have to reverse engineer the document using the harmonized Q6A framework. Returning to the master specification table, we isolate the top section. These 4 parameters, description, identification, assay, and impurities, are the universal tests. They are required for every new chemical entity, regardless of the dosage form. The mandate for identification testing is absolute specificity. The test must accurately discriminate the active ingredient from closely related compounds. Therefore, relying solely on a single chromatographic retention time is invalid. Manufacturers must deploy orthogonal methods to achieve acceptable specificity. Moving to impurities, the regulatory framework introduces a strict bifurcation. We must separate organic impurities arising from the drug substance synthesis from those arising as degradation products in the final manufactured drug. The Q6A rule dictates that process impurities from synthesis are controlled during drug substance testing. They are excluded from the total impurities limit of the finished drug product unless that specific synthesis impurity is also a degradation product. These universal tests form the non-negotiable baseline. They legally guarantee the basic identity, purity, and strength of the active ingredient before any complex formulation-specific variables are introduced to the control strategy. Below the baseline, we find the specific tests. These parameters are not universally applied. They are dictated entirely by the drug's physical chemistry and the chosen commercial dosage form. If a new drug substance exists in multiple crystalline forms affecting bioavailability, the manufacturer must establish stringent solid-state specifications. Since measuring polymorphic changes directly within a finished drug product is difficult, Q6A favors surrogate tests, like dissolution, to monitor performance. For immediate release solid oral dosage forms, dissolution testing sets the baseline expectation. Measuring the precise drug release rate in vitro ensures consistent in vivo performance across every manufactured batch. This logic tree charts the exception for highly water-soluble drugs. If dissolution is consistently greater than 80% within 15 minutes across the entire physiological pH range, the regulatory framework allows manufacturers to swap complex dissolution protocols for simpler disintegration testing. Specific tests are never arbitrarily assigned. They are precisely mapped to the drug's physical vulnerabilities, scientifically guaranteeing the formulation's clinical robustness under commercial manufacturing conditions. Specifications also provide flexible frameworks that allow manufacturers to reduce their testing burden, provided they have the empirical development data to justify it. We see this heavily in microbial limits and assay acceptance criteria. This introduces the concept of periodic or skip testing. For attributes like microbial limits in solid oral dosage forms, manufacturers can perform specified tests at predetermined intervals rather than on a batch-by-batch basis. To implement skip testing, the manufacturer must provide robust historical development data proving the product formulation simply does not support microbial viability or growth. This graph illustrates the strategy of release versus shelf life acceptance criteria. A manufacturer may establish a tighter in-house assay limit at the time of release. This provides increased assurance that the product will remain safely within the wider regulatory acceptance criterion throughout its entire shelf life. How this strategy is handled depends on regional harmonization. The European Union requires distinct regulatory specifications for release and shelf life if they differ. In contrast, regulatory authorities in the United States and Japan treat tighter release criteria strictly as an internal safeguard. The Q6A framework actively rewards manufacturers who possess a deep understanding of their process consistency, offering operational flexibility in exchange for rigorous data. With the required tests defined, the final step is setting the hard numerical limits, or acceptance criteria, located in the right-hand columns of the specification table. This scatterplot shows how these exact limits are derived. The acceptable ranges are not theoretical. They are established by plotting actual development batch data alongside results from accelerated and long-term stability studies. At the initial time of regulatory filing, the amount of available data is naturally limited. Because process consistency at a massive commercial scale cannot yet be fully proven, initial acceptance criteria must focus strictly on broad boundaries of safety and efficacy. As commercial manufacturing scales up and process consistency is proven post-approval, Q6A expects these initial limits to be continuously reviewed. Depending on the accumulated data, this results in either tightening or loosening a criteria over the drug's lifecycle. The specification is a living document, dynamically shaped by the physical nature of the molecule and the practical realities of commercial manufacturing scale-up. Once finalized, this document serves as the definitive legal and scientific handshake between a pharmaceutical manufacturer and global regulatory authorities, ensuring every unit on the market acts exactly as intended.